Hello, everyone. My name is Carolyn Cassady. I'm an analytical chemist. I want to tell you a little bit about my research involving mass spectrometry. Now, mass spectrometry has been the fastest growing area of analytical chemistry in the past 25 years. You could actually argue that it has been the fastest growing area of chemistry. Mass spectrometry now does so many different things. A major emphasis is mass spectrometry of biomolecules. My group works primarily with peptides and proteins, but we also work with carbohydrates, which are also known as glycans. A major emphasis of our research involves peptide sequencing by mass spec. This is an established technique, but it still needs a lot of development. It's not as successful as it might be desired. Now, peptide sequencing by mass spectrometry is the basics for proteomics. This is determining the amino acid sequences of the body's peptides and proteins. This is important to the diagnosis and treatment of diseases, for example. In COVID-19, the first step to developing a vaccine was actually to sequence the peptides involved in the virus so that the vaccine to target this could be developed. Now, of the routine methods involving mainly protonated positive ions as sequenced in mass spectrometry, they're not actually as successful as you might expect from the literature. For example, a survey of 162,000 yeast peptides sequencing with mass spectrometry only identified 27,000, which is about identification yield of 17%. Other studies had an identification yield of even less. So there is still a great deal of work to be done to improve peptide sequencing by mass spectrometry. Now, the vast majority of sequencing does involve the protonated positive ions, which are made by taking a basic site on the molecule and adding a proton, using electrospray ionization or MALDI, matrix-assisted laser desorption ionization. What my group does is we work with other precursor ions. We just change the ion we're dissociating with, which does not involve a costly instrument modification. It just involves developing your techniques on your existing mass spectrometer. We have three focuses. We're focusing on looking at deprotonated ions, negative ions, which is especially useful in acidic peptides that will prefer to deprotonate rather than protonate. We're also looking at metallated peptide ions, where in place of adding a proton, you add a metal ion. Plus, one of the things we discovered while doing our metallated peptide work is that some metal ions will actually enhance the protonation, will cause protonation to occur. This process is known as supercharging. We use a variety of tandem mass spectrometry techniques. My research group has a Bruker BioApex electrospray ionization Fourier transform ion cyclotron resonance mass spectrometer. We can do ion molecule reactions, which to determine gas phase acidities. We can do collision-induced dissociation. We can also do electron transfer dissociation or electron-induced dissociation. The University of Alabama Mass Spec facility has 6 mass spectrometers. I'm the faculty supervisor of the facility, which is in Shelby Hall on the first floor adjacent to my laboratory. Our newest instrument, which we had a little bit over 2 years, a Bruker Rapiflex, can do post-source decay, in-source decay, and high-energy collision-induced dissociation. We also, of the 6 mass spectrometers in the facility, another one that my group uses extensively is the Bruker HCT Alter. It's an electrospray quadrupole ion trap mass spectrometer. It can do CID, it can do electron transfer dissociation. That is a major strong point of this instrument. There's also a very nice UPLC LCMS type system, a Waters ZBO system in the QTOF in the mass spec facility that my group occasionally uses. This is a picture of our Fourier transform ion cyclotron resonance mass spectrometer. You can see an electrospray ionization source, ion transfer, lots of pumping to move the ions into the superconducting magnet. This is a 7 Tesla superconducting magnet. Our Bruker Rapiflex, which arrived in 2018, late 2018, 2019 actually. You put your sample on a plate here mixed with a MALDI matrix. You blast the sample with a neodymium mag laser. The ions are accelerated up the flight tube. There's a collision chamber right about in here to break the ions apart and look at the fragment ions. Then they travel about a 3-meter path over to the detector that's over in here. This is a state-of-the-art tandem TOF, a MALDI TOF. It is the top of the line instrument, an extremely nice instrument that's available to any research group on campus to use. Now, a focus of our work is tandem mass spectrometry. In tandem mass spectrometry, you take your sample, you do ionization by a technique such as electrospray and MALDI. You use your first stage of mass analysis to separate out your precursor ion, which you're going to then dissociate. We use techniques such as collisions and collision-induced dissociation. We use electrons into electron transfer dissociation. We perform ion molecule reactions, something to either dissociate or react your precursor ion and make product ions, which you then mass analyze in your second stage of mass spectrometry. And from there, you detect these product ions. And from this information, you can get a molecular weight information on the sample from the first stage. And from the second stage, you get structural information about the sample. Now, some of our projects involve work with peptide negative ions. This has previously been funded by the National Science Foundation. It's a joint project with Dr. David Dixon's group at the UA Department of Chemistry, where the Dixon's group does calculations to support our mass spectrometry experiments. A big thing we have been focusing on is measuring gas phase acidities of small peptides. This involves the ion molecule reactions in the FTICR, and in conjunction to help us interpret the results, the computations from the Dixon group. We've already also spent a great deal of time looking at trying to elucidate the deprotonated peptide fragmentation mechanisms. That is, determine which bond is cleaving experimentally, why is it cleaving, what structures are being formed. In addition, a big thing that we're working on right now is adding electrons to these negative ions, which is not an easy thing to do, but this results in negative electron dissociation techniques, EXD type of techniques that can give sequence information for the peptides. So what my group does is we develop the fundamental information of which are the best processes, what are the energetics of processes, what are the structures. We get the fundamental information that people sequencing real-life peptides can use in their experiments. We also are in the process of developing high resolution, high energy CID on the RapidFlex for the development of sequencing deprotonated peptides, acidic peptides that want to make negative ions. This is an example of a spectrum in our RapidFlex to the MALDI TOF instrument. It's called in-source decay. This is the MALDI matrix. One of the important things about the MALDI experiment is to determine the best matrix to use. And in this case, we want the peptide ions to dissociate in the ion source. This is called in-source decay. And this is a matrix we have found that imparts energy directly in the source. So it makes fragment ions here. So insulin chain B, our analyte here, has 30 amino acid residues, and you want to cleave between each and every one, and it pretty much does a very good job of that. So it's a very useful technique for peptide sequencing. Especially, we're the first group to have ever done and published a paper on this, just dealing with the negative ions. Also in the RapidFlex, it has high resolution, which involves especially good mass accuracy, high resolution with high collision energies. And there are very few instruments that can do this. This is looking in a peptide in the positive mode and the same peptide in the negative mode. What you want is it to cleave between every residue. You can see this cleaves between these residues and this cleaves between these residues. So taking the positive mode and the negative mode together for the same peptide, you can cleave between all residues. This makes it especially useful for peptide sequencing. There have been no previous reports that have used high resolution and high energy in the same experiment. We're working on our first manuscript on this. It will be the first report of its time. Another thing that we've been doing, and that has been in the past has been funded by the National Institutes of Health, is rather than adding or subtracting protons, we've been adding metal ions. Such we've added the lanthanide metal ions, we've added many transition metal ions. We've looked at the identity of the metal ion, the charge state of the metal ion. We're especially focusing on electron transfer dissociation, which we can do in our HCT Ultra quadrupole ion trap LCMS. And we found this is especially useful for metallated peptide ions. It provides a great deal of sequence information. We also recently published a paper using it for methylated glycans or carbohydrates, where it works very well. This is, for example, the lanthanide metal ion presidonium. We did the whole lanthanide series and found presidonium to work especially well. This is working with a peptide for brenopeptide B, +2, +3, and +4. And the +4 ions are especially useful. In general, the higher the charge state, the more fragmentation you're going to see. This +4 ion, it cleaves at every single residue. This is what you want for peptide sequencing. In fact, I think that sequencing peptides with electron transfer dissociation on metal ions is the very best method of peptide sequencing. It's better than what people are doing now. Now, one of the things that we noticed with our enhanced protonation with our metal ion studies is that in some cases, the metal ion would actually increase the protonation. And this is what the focus of our current National Science Foundation grant, which is a project in collaboration with the Dixon Group, who does calculations, and the Vincent Group, who is an expert on chromium, because chromium-3 is the best metal ion to do this. We've been studying the conditions for this enhanced protonation. We've also been studying the mechanism. We actually last year published a paper on the mechanism, but we're still working on that. For example, where is this extra proton, this enhanced proton that the peptide is taking? Where is it going? How is it getting to the peptide? We're working with it on mixtures with LCMS, and we're also looking into whether other compounds can do this process as well. But let me show you what I mean here. This is called chromium binding peptide. It's just the first peptide we worked with. It's a very acidic peptide. It has small peptide. It has glutamic acids, a glycine, and 2 aspartic acids. So very difficult to make a positive ion signal by protonation with electrospray. This is, for example, the signal without chromium. What you need for electron transfer dissociation experiments is you need doubly charged ions. This is a doubly charged ion here. And there's not enough of it to work with at all. It's just too weak in intensity. But you add chromium, and suddenly this ion just pops out, even though what it did was added another proton. In fact, our paper last year on the mechanism of this shows that it really, when you have chromium hexahydrate, where chromium 3+ in solution has 6 waters around it, as the waters strip off during the electrospray drying process, it turns the chromium ion into a superacid that can transfer protons from the water. And we found that it can do this for many other peptides as well. This peptide ion is now intense enough to study. We do electron transfer dissociation on this peptide. It cleaves at every single residue, and it allows you to sequence the peptide. This is a peptide we tried to sequence with the Vincent Group for years before we discovered this technique, and it was very difficult to sequence because of its high acidity. But now it can be sequenced readily. So that's all I have to tell you for today, but I would be happy to answer any questions from you by email. Just send me or anyone else in the department any emails if you're interested. We'd be happy to talk to you more. I hope you have a good day.